Great, thank you for the introduction. So I'm very excited to be with you here today to talk about the quality considerations for transdermal systems, or TDS. So before we get into that, first, what do I mean when I talk about pharmaceutical quality? Well, you might recall that pharmaceutical quality, we're talking about a quality product of any kind consistently meets the expectations of the user. So drugs are no different. Patients expect safe and effective medicine with every dose they take. So pharmaceutical quality is assuring every dose is safe and effective, free of contamination and defects. It is what gives patients confidence in their next dose of medicine. So transdermal products are no exception. So to begin with, I'll kind of talk about the quality aspects of a TDS, and then I'll focus specifically on the agency's expectations for extractables and leachables, what happens if you need to change a component prior to approval, adhesive quality tests, and then some labeling considerations from the quality side. So to start with, like with any pharmaceutical product, we want to design with the end user in mind. So TDSs have, specific quality concerns, as Dr. Ghosh spoke about, such as adhesion to the skin, irritation, different safety aspects, patient use aspects. So all of these then correlate to unique quality aspects. So what's bolded here are the sort of the aspects that I'll touch upon in my talk. For some of the other aspects, I'd refer you to my colleague's talk in this forum from 2018, and there's a link in your slides. So first, let's look at a TDS. So again, Dr. Ghosh showed a lot of different designs, so I'm going to focus on the most simplest design. Let's keep it simple. So here's a matrix design, and there's the release liner. So this is what's actually removed by the patient prior to administration, and generally this is a PET material. And then there's the backing membrane. So after the TDS is applied, this is what will actually be facing out. So this contains, this is made of different polymeric materials, and this contains a printed identifying label with the drug name and strength. And then there's what is actually adhering to the patient, the adhesive layer. And this contains drug either in a dissolved or dispensed state, dissolved or suspended state. And there may be different types of adhesives. You may use one or more different grades. And there may be additional excipients in this layer, such as permeation enhancers. And so again, this is just a simple design. Your design may also incorporate multiple adhesive layers or an internal membrane. So let's take that transdermal system and let's look at it on the side, sort of like a sandwich. And so that brings us to our first quality aspect, which is extractable and leachable studies. So why are these important? First off, they're needed to demonstrate drug product safety. So when we think about the different components that I just outlined, there could be impurities in these components, and these are called leachables, and they could be entering into the adhesive matrix, such as from the pouch, the backing membrane, or the release liner. So any impurities that are already present in the adhesive matrix, those are not what I'm talking about when I talk about leachables. So those are out of scope for what I'll be talking about next. And so, in addition to safety, why else is this important? Well, a lack of extractable, leachable, or EL studies, or inadequate EL studies, may result in a major deficiency. You don't like that, and we don't like that. And so you can refer to this guidance for more details. So, if you want an adequate EL study, what are we expecting as an agency? So that's what I'm going to go over next. So first in the extractable study, so let's begin with the goal. The goal of this study is to identify potential leachables that could end up in your drug product, and then demonstrate that you have analytical methods that can detect these potential leachables. So I'm going to kind of go through some different critical elements and what we're expecting. So first, you need to start by pre-identifying the potential leachables. So when you go and get your component from the vendor, they should provide you information on what kind of impurities are present in this material. And then you can do an analysis of, based upon that information, which of these impurities are most likely to leach into your drug product. And then based upon that, you can design your extraction conditions, and we would expect you use several solvents to cover a wide range of polarities, and that you're justifying extraction time, temperature, volume of solvent, the amount of the component that you're extracting. And such a justification might show that you're getting asymptotic levels. As you measure over time, you're getting all the extractables out. And then you're also comparing back to your pre-identification work and saying, yeah, we're getting out what we expected to see. And then as far as the analytical methods, we would expect you to use multiple methods so you can detect volatile, semi-volatile, and non-volatile organic compounds. And then you're designating these extractables per USP 1663 guidelines. So that brings us to the next study, which is the Leachables study. And so the goal of this study is to identify and quantify any leachables present in the adhesive matrix at release and during stability, and you're using defined analytical thresholds for this. So I'm just going to compare and contrast some hypothetical inadequate leachable study with an adequate leachable study. And so these are just examples. They're not all encompassing, but I think you'll start to get the idea. In the inadequate study design, maybe you don't remove the release liner when you expose it to the solvent, where you do remove that for the adequate study. In the inadequate study, maybe you're just using water as your extraction solvent, where in the adequate study, you're using an extraction solvent that mimics the worst case clinical conditions of the skin, such as a simulated sweat solution. In the inadequate design, maybe it's just sitting there with no agitation, where in the adequate design, you have it on a shaker to try to improve the extraction. And then, maybe you have no demonstration that your levels are reaching an asymptotic level for the leachables in the inadequate study design, where for the adequate study design, maybe you're providing that as justification for your conditions. So moving into some of the other elements, the analytical methods, generally, these should be the same as what you use in the extractables study, because after all, that was one of the purposes of that study, to show that you had suitable methods. We do want to get kind of an idea of the variation that you might see in leachables in your product. So we would expect to use at least 2 exhibit batches for this study. And then also, we would also expect you're looking at multiple time points for your leachable study. So why is that? Let's look at this hypothetical leachable level over time. So these tend to be reactive components. They might degrade further to different components. So you need to really look at a couple different times to kind of get an idea of the maximum exposure of this impurity in the product. So, you have your studies, they're designed well, and you've identified, and you want to say, okay, how do I identify these leachables? Which ones do I need to be concerned about? And that is defined based on the safety concern threshold, and that is product specific. And so that is, translated, so to speak, into an analytical evaluation threshold, or AET. And so you want to make sure that your limit or your methods can detect at a low enough limit to the AET at least. And then anything that you're detecting above the AET, you need to identify and provide a structural analysis of that compound. So for your reference, here's just a general way to convert from the SET to the AET for a transdermal system. So then, any of these leachables that you're detecting above the SCT need to be qualified for safety, considering both local and systemic toxicity. And inadequate qualification will result in a major deficiency. Again, we don't want that. So for your reference here, when you're thinking about developing these products, these are SCT values for different marketed TDS that have been determined by my colleagues in OGD. Okay, so that moves us to the second quality consideration, and that is what happens if I need to change a component prior to approval. So there could be a variety of reasons for this. Maybe the vendor's not making it anymore. Or maybe you notice on your stability batches that there's some quality concern and you think it's linked to that component. So a lot of times maybe we see changes because of a pouch stock needing to be changed or a release liner. So I'm going to walk through some different examples for that. But before we get into those, I wanted to first define cold flow, because that will come up in the examples. So cold flow is defined as the movement of the adhesive beyond the edge of the TDS or between the slit in the release liner. So if you kind of look in the schematic there, maybe you see some adhesive coming beyond the edge of the system. Or in the photo there, if you were to trace a line around the boundary, you see some adhesive has come past the edge of the backing membrane. So this is a quality concern and a patient use concern. So first off, if you have a lot of cold flow, the patient might not be able to pull it out of the pouch. It might be stuck inside. And then also, while the patient is, after administration, if there's cold flow, maybe it might stick to something and then it falls off. And so then that also poses risks to maybe inadvertent exposure depending upon the drug. Okay, so that's cold flow. So now let's look at example 1. And so in this, during development, the applicant had noticed that there was a lot of cold flow from their product, especially coming from the release liner slit. They were using this S-cut kind of design. So they proposed changing to a design with no release liner, where instead there would be a notch, and the patient would just tear the release liner and then adhere the product on that way. So there was, a slight concern that this is a different type of release liner design. Maybe the patient might not know what to do with this. So the applicant was requested to add a second notch and to make the notches more apparent. So then the question is, okay, what sort of quality information do I need to provide to support this change to the release liner? So in this case, we just need a diagram showing the differences between the exhibit batch design and your commercial design. So, just simple documentation. Okay, so now let's look at example 2, also involving cold flow. So in this case, the cold flow was noted during the stability studies, for the exhibit batches. And so in this case, the applicant had designed a product that had dimples in the release liner. And those shown on those circles on the release liner there. And so that's thought that, during storage, as these are in the carton, that then they don't press against each other as much and they don't, sort of squeeze the adhesive out. So the applicant proposed, we'll take these dimples and we'll put more of them and we'll increase the size of them. And then we're also going to move the release liner slit so less of the system is exposed to that, less potential for cold flow to squeeze out that way. So in this case, then, what quality information did we expect? So since this was, an issue noted on stability, we would want to see an additional exhibit batch manufactured with these changes, and then look at the accelerated and long-term stability data to show that, yes, these changes did actually mitigate this cold flow concern. And then, with this exhibit batch, we would expect all the quality documentation like batch records. So then let's look at example 3. In this case, there was high release liner peel. And that poses a risk to patient use because the system may be hard to remove from the release liner, or during removal, as is seen here, there may be adhesive transfer, where some of the adhesive ends up on the release liner. So in this case, the applicant saw this increasing trend of release liner peel force during stability, and they proposed changing the release liner to a new material to address this. So the quality data, again, we need to see an additional exhibit batch with the stability data that shows this sort of trend is no longer occurring. But now you've introduced a new component, so we do need extractable, leachable type studies to support this new release liner. And that brings us to sort of a general concept here, which is anytime, let's say you have a product, you've qualified all the components with extractable, leachable studies, but then you need to change a component. So in this case, we would advocate a risk-based approach, where first you look at a comparable extractable analysis between the new component and the old component. And if they're similar, you're seeing similar things come out in the extractable study, then that's a lower risk. Where if they're different, then that would be a higher risk. You're seeing new extractable components maybe. And so then based upon that risk, you can decide how to proceed to qualify this new component. All right, so that brings me to my third quality consideration, and that's adhesive quality tests. And these are tests that are performed for quality control. And so there's peel adhesion tests, release liner peel, shear, and tack. And what are we looking at as the agency? We're using this information from these tests to ensure consistency, both within a batch, between batches, during stability, and between commercial batches and those original clinical batches. So I'd like to focus for the next couple slides on peel adhesion. So peel adhesion has an ASTM standard for this method, and there's two kind of ways you can do it that are shown here. So the first way, in that method A, on the TAXT plus, that's a 180-degree peel from steel. So in both of these cases, you have the system and it's been adhered to a test panel. So in method A, you're pulling at a 180 degrees from the test panel, and you're measuring that force of removal. And then in method F, that's shown on this Chem Instruments AR1000, then you are pulling at a 90-degree angle. Same way you're measuring the force that it requires to remove the system from the test panel. So these are quality control methods, so precision should be demonstrated by method validation. And the maximum variation that you would expect to see is also discussed in that ASTM method. But variability, as much as possible, should be minimized as you develop the method to make sure this is a suitable quality control tool. And I'll give you an example of that. So let's say I see this data of peel adhesion during long-term stability. I'm thinking trend. Maybe the applicant is saying, no trend, this is due to analytical variability. Well, if your method can't sort between a trend and stability or analytical variability, then it's really not suitable for use as a QC method. So if this sort of arises, you might see a deficiency such as this be issued by us, where really the punchline here is we're going to recommend you modify the peel adhesion method to reduce variability in the results that you're reporting. And there's a lot of places where variation can come into play. So this is for your reference. I'll just highlight on sample preparation. So you'll have the system, and maybe you're cutting it to a specific width with specimen cutters. And then you're taking it, you're adhering it to the test panel with a roller. And that roller has a specified weight that you've checked and calibrated. And then you're rolling it multiple times. Is it all in the same direction, or are you rolling back and forth? All of these things can be specified in your method, and that will minimize sources of operator variation, which will in turn minimize sources of variation in your data. And so then once you have this method, you need to set acceptance criteria for these tests. And in general, for all of these adhesive quality tests, we expect acceptance criteria to be a range with a minimum and maximum value. And we are expecting that the acceptance criteria is justified by the results for the clinical batches at release, and not justified by looking across the variation and all of the stability data for all of the batches. So again, let's look at this as an example of why that is the case. So if I look at that release value times 0, I might set a range such as this. Okay? But then let's say, well, I'm going to consider all of the data I see on stability. So that gives me a much wider range for my acceptance criteria. But then that will lead to the question, what about these time points out here? There's no assurance that the peel adhesion values out here will result in similar product quality to what we saw at release. And so that's why we really would advocate that you look at the clinical batches at release when setting these values. Okay, so then briefly I'll just cover some of the quality considerations we look at for the labeling. So for transdermal products, right, there's labeling right on the product itself. It has an identifying label on that backing membrane. And some of the expectations for that are outlined in this guidance for your reference. But basically, the identifying label should list the drug name and strength. It should be easy to read. It should be durable. And then for specific products, there may be certain requirements like fentanyl due to safety reasons. So, speaking to the durability, if I have a system, and I go to adhere it, and maybe I rub off the identifying label, that could pose a concern, especially if the product has, a certain safety consideration. So if you see, where you're having a problem with your ink, then maybe you might need to change the ink. But that links back to the first quality consideration, right? And the second quality consideration. I'm changing a component now. I need to qualify the addition of that component, potentially by extractable leachable studies or ink penetration studies. So better to, evaluate this aspect first before you get to this point where you have to change it. And then also, in the description section of your labeling, you should be listing all inactive ingredients, and that does include the release liner and the backing membrane. And then finally, as a generic, you're, generally making your labeling match the RLD. But because your product might have a different design, right, it doesn't need to be Q1, Q2. With the RLD, you could have a different shape, a different release liner design, a different identifying label on there. So the instructions for use in the labeling should accurately reflect your product's design and not just copy the RLD's information over there. So then just to conclude, very important that adequate extractable and leachable studies are provided. So we need these before approval and also maybe to support some changes to the product later on. And so component selection and design are critical to achieving the quality that you want in your drug product. So if you see that you have to change a component to get that quality and you're, later on in the process, then, there's various degrees of quality data you'll need to submit to support your changes. Adhesive quality tests are a quality control tool and should be, developed and validated as such so we have reliable data. And the labeling should accurately reflect your generic product. And then finally, the identifying label on the system itself should always include the drug substance, name, and the strength. So with that, I'd like to acknowledge my colleagues in OLDP and the CDER Transdermal Working Group. I'd definitely like to acknowledge Anna Wokovich, who provided the pictures of the adhesive test equipment and tools that you use for that. And also would like to acknowledge my colleagues in OGD, which were instrumental in developing our approaches to extractable leachable assessment. And so with that, I thank you for your attention, and I look forward to your questions on the panel.